as I've said earlier, because you have 6 billion base pair in your genome, it will happen, there will be some mistake made. And the cells is well done because you have some mechanisms with genes, which are called DNA repair genes, that will come and repair these genes. So you'll have a signal, P53 will send a signal that the cell needs to stop to divide. Then there will be some proteins coming to repair the DNA, and then the cell will be able to restart the cell cycle and divide. There will be also some stress that will come, and this, again, P53 will tell the cell to stop the cycle, repair the damages, and then restart the process. So when these damages are minor, P53 out the cell cycle, but then if there is really big damage and it cannot be repaired, and that will happen, P53 will trigger the cells to commit suicide by apoptosis. This is quite a complex slide, but it's just to, it's for you to see that P53 is really a crucial tumor suppressor gene in cancer, and not only in familial, but also on spontaneous cancer. And lots of different types of cancer have mutation in P53. And P53 is important because it's important for DNA repair, for growth factor, senescence, or disease. There is many ways in which P53 may malfunction in human cancer. Here is a few examples. You can have some amino acid change and mutation in the DNA binding domain. So P53 bind to other genes because it's a transcription factor. So it binds to DNA to regulate expression of certain genes which are involved in apoptosis or involved in cell death. The typical tumors that are associated with DNA changes in P53 are colon, breast, lung, bladder, brain, pancreas, stomach, and many others. So it's just an example to show you how P53 is really an important tumor suppressor gene in human cancer. And there's lots of different mutations that can happen in P53. So I've explained to you what is a tumor suppressor gene, and actually tumor suppressor genes are involved in what we call genetic instability. And tumor suppressor gene can be even more defined as either gatekeeper or caretaker. So what's a caretaker? A caretaker, it protects the genome against mutations. So DNA repair genes, BRCA1 and BRCA2, they are tumor suppressor genes, but they are caretakers. So they are involved in genetic instability. And you have genes like P53 or APC, they are gatekeepers because they induce cell death or cell cycle arrest. And in this cartoon, it actually shows you that if you, in your germline cell, you already had a mutation in one of these gatekeeper genes like P53 or APC. I'll give you an example of germline mutation in APC. If you have also a mutation in the caretaker genes, it will create an environment where you have genetic instability. When you will start to acquire more mutation, and then since your gatekeeper already had a mutation because you inherited that from parents, it will only take one other hit for tumor to initiate and progress. So DNA repair, it's any one of many cellular processes that attempts to correct errors in cellular DNA introduced either via the environment. It could be, for instance, smoke from cigarettes, it can be UV radiations, and so on. Or it can DNA can be, errors can come from during the cell division with the polymerase, for instance, who makes errors. DNA repair genes are mutated, and cells cannot repair DNA to conventional oncogenes, which cause accumulation of the malignant characteristics. DNA repair defects, mismatch repair and microsatellite instability. So repair DNA replication errors, these genes, this mismatch repair, they are defective. These genes, MMR is the mismatch repair genes, they are defective in certain cancers. So for instance, I'm going to give you an example later of some type of colon cancer, and that will induce a microsatellite instability, and I will explain what is a microsatellite. There is some germline mutation in these MMR genes, and they are associated with some type of colorectal cancer, and one of these colorectal cancers is called HNPCC for hereditary non-polyposis colorectal cancer. But these same genes, they can also be involved in sporadic cancer. And the type of sporadic cancer they are involved in can be also colorectal cancer, but it could be also endometrial or gastric cancer. So what's microsatellite instability? So in the DNA, you have some repeats of 1 to 6 nucleotides, and that's normal. And your mom may have, for one particular piece of DNA, may have 10 repeats, but your dad can have 8 repeats. So there is the length of this DNA, of these repeated DNA sequences is variable, and it will change between individuals. And as an example, often it's a CA denucleotide repeat. So you can have it 6 times, 7 times, 8 times, 9 times. And if you have some errors in this, and you have a 5 branches errors, it can be implicated in some cancers. Another, I think you're going to have a lecture next week on apoptosis, on cell death. There's also some mutations that prevent the cells from undergoing apoptosis, and then can allow the cells to accumulate and prevent elimination of cells with mutations to other genes. And so apoptosis is also called programmed cell death, and there's different pathways. There's an extrinsic pathway and an intrinsic pathway. The intrinsic pathway goes through the mitochondria, and the time to completion is usually around 24 hours. And there's lots of different molecules which are involved in apoptosis. For instance, mostly some caspase. I believe you'll have a lecture next week on apoptosis. But what you need, the take-home message here is that you can have mutation in either oncogene or tumor suppressor genes will have an impact on apoptosis. There's also another biological process important in cancer is cell cycle. So most of the cells, when they divide and during the replication, then some errors occurs. So then to repair these errors, you have checkpoints in the cell cycle, and to ensure that the DNA that will be replicated will have new mutations, and then you can have the cells that can divide. So you have some really important tumor suppressor genes which are checkpoints of the cell cycle. And this is an example of these genes. P53 and ARB that I've talked to you before, they are involved in checkpoints on the cell cycles. And you have also oncogenes like MYC, the MYC oncogene, or the RAS oncogene. When I showed you the cell signaling, and when I showed you that there was some signal going through the cell, through the nucleus, RAS is one of these signaling molecules. So there's different types of genetic changes. It's either chromosomal changes, where you have a gain of material, so it can be like, for instance, amplifications, or oncogenes are known to be amplified. Or you can have loss of material, where you lost one of the allele, and usually it's for tumor suppressor genes. Or you can have a relocation of genetic material without a net gain or a loss, which is called a translocation. And I'll give you an example of translocation. So that's at the chromosomal level, but you can have also mutation that occurs at the DNA level. So in sporadic cancer, it's going to be a somatic mutation, and if it's a germline mutation, then it will cause hereditary cancers. But there's not only genetic mutations at the DNA level that change the nucleotide, or small deletion or insertion. You can have also what we call epigenetics. And epigenetics, it can be the promoter methylation or histone modification. But I'll explain later on more about epigenetics. So chromosome rearrangements is changes in chromosomes that put one next to the other one. You know, I've showed you a slide before with the 3 types of mutations that can occur in oncogenes. So then you have one gene that was not necessarily expressed who becomes all of a sudden expressed because it has changed its promoter. Or you can have also gene duplication where you have extra copies of the same gene. And so then you have too much of a certain protein. So that's an example of chromosomal translocation in tumourigenesis. So you have your cell, and you see you have down the chromosome red that has exchanged some genetic material with the chromosome yellow. And that gives rise to a chimeric protein or a fusion protein that will have part of the yellow and the red. And this will, this new protein, because it's basically a new protein, will alter the gene expression of lots of other genes. So for instance, MYC, which is an oncogene and is a transcription factor, if MYC is fused to another gene, MYC is expressed all the time. MYC is not supposed to be on in normal cells, but if it becomes expressed all the time, then it triggers some expression of genes which will cause cancer. That's a table. It's just for you as an information. It's all of the different types of translocation. And usually translocations are found in hematological disorders or in solid tumors, but mostly sarcomas. So far we haven't really described most translocation in other types of solid tumors like lung cancer or breast cancer. Although now with new technologies like next generation sequencing, we are discovering more and more new type of mutations, and some of these solid tumors like lung cancer or prostate cancer, they have translocation. But here the take-home message is like you find translocations mostly in hematological disorders, in leukemia or in lymphomas, and in certain types of sarcomas, which are rare disease. So an example of translocation it's the Philadelphia chromosome, and this is a translocation that has been found in chronic myeloid leukemia. It's a translocation of material between chromosome 9 and chromosome 22, and that gives rise to a gene fusion, BCR-ABL1, that produced an abnormal tyrosine kinase. So again, this is related to cell signaling. Tyrosine kinases are these molecules that transmit the message to the nucleus. And in that case, these two genes are fused together, and that produces a new protein that will have oncogenic properties. And actually, that's an interesting example for 2 reasons. One is you'll find out that, I've told you about Bovary, who discovered that it was one single cell had a genetic alteration, and then tumor is a clonal disease. All of the cells in chronic myeloid leukemia, they have this translocation, this Philadelphia chromosome. And the second thing why it's an important disease is that the fusion protein, there's actually some drugs now which target this fusion protein. So that's a really good example of translocation. Another example of translocation in hematological cancers is the Burkitt lymphomas. And again, it's one of these oncogenes MYC that I described before. In the normal chromosome, it's on chromosome 8. And you have this immunoglobulin genes on chromosome 14 that is fused in lymphomas to MYC. So if you know about human cells, they have to produce all of these antibodies. So they have to be constantly expressing the genes to produce antibodies. So if you put the promoter of one of these immunoglobulin next to an oncogene, then you find out that this oncogene then is going to be turned on all the time. So you know when I told you about the car with the accelerator and the brake, MYC all of a sudden is expressed all of the time because it's being fused to this immunoglobulin that drive the expression of MYC. Loss of heterozygosity. So to go back to tumor suppressor genes, when I told you about the mechanism to hit, you need to have 2 mutations in a tumor suppressor gene to drive cancer. So you usually have mutation in one of the allele, and then you need to have a mutation on the second allele. And most of the time, it's a deletion. So well, I'm explaining correctly. So when you have a germline mutation, you only have one copy of your allele that is mutated. And for this tumor suppressor gene to be inactivated, you need the second allele. And the second allele will be deleted, but that will be in the somatic cells. So for instance, to take the example of breast cancer, you have one of your allele for BRCA1 or BRCA2, which is already mutated, and then you have a second mutation, which is a loss of the other allele that happens. But that won't happen in your germline cells, because it will happen in your somatic cells and it will happen in the breast cells. Another way as well to mutate a gene is epigenetics. And actually epigenetics is defined as inheritable changes in gene expression, but unlike mutations, it's not attributable to an alteration in the sequence of the DNA. So epigenetics is more related to the regulation of the genes. And the regulation of the genes can be either at the level of the promoter, when you find that you have hypermethylation of the promoter, so you can't have some factors or proteins that binds to the promoter to turn on your genes. Or you can have modification of the chromatin, or you can have also non-coding RNA that will regulate the genes. That's exactly what I've just explained. You can have CPG island is a region where you have the promoter, and in cancer you can have this promoter that is hypermethylated, so the genes won't be expressed. So a tumor suppressor gene has a normal function to inhibit cell growth. So you can imagine that if a tumor suppressor gene is not expressed, then it won't be able, for instance, to read your DNA, or it won't have this function to stop the cell cycle. So it's really important that tumor suppressor genes are expressed in normal cells because they inhibit cell division, or they read your DNA. Are you okay? Yeah. Do you have a question?
So in what conditions these tumor suppressor genes are not expressed?
Sorry, say it again?
In what condition? Like when are they not expressed? The tumor suppressor genes.
So the tumor suppressor gene, it depends if it's in the sporadic context or in the hereditary context.
So is there a possibility that a person has a gene of cancer?
Yeah.
Either somatic or germline. So is there any possibility that the suppressor gene has not been expressed in order to inhibit cancer?
When you acquire the mutations, there is no way back. So if you have a tumor suppressor gene that is mutated, it's irreversible. So if it happens in the genes that